Today I'm going to talk about how we came to discover that we could use an old PCR machine to rear my study organism, Ceratina calcarata. So we've heard a little bit about Ceratina already today, but just as an overview, I'm studying C. calcarata, which is a native bee to eastern North America. It's a small carpenter bee that nests in twigs and canes of raspberry, rose, sumac, and teasel. Its choice of nesting substrate makes it incredibly easy to experimentally manipulate and observe. We can do this by attaching a raspberry cane to a stake and putting the cane out in a sunny spot near some natural raspberry or sumac in the spring. And immediately in the spring, you can attract females who will start to dig new nests. So in the bottom right-hand photo, you can see a new female digging a new nest, and then these are some twigs that I attached to the bamboo stakes. And once the female's inside, either in the early morning or in the evening, you can move the nest to a new experimental location. It's important to note that the mother's choice of nesting substrate and location can naturally affect the microclimate in which her offspring develop, but we can also move the nest to experimentally affect that microclimate ourselves. So for my master's project, I'm investigating the effects of developmental temperature on Ceratina calcarata physiology. For today's talk, I want to focus on the portion of my project that looks at the effect of temperature on the development of C. calcarata offspring. My objectives were to determine the temperatures that developing offspring experience in the field, and then to examine how different temperatures affect offspring development time. So just as a bit of background, C. calcarata are ectotherms, meaning that they can't regulate their own body temperatures, and so their physiological processes, like their metabolism and their developmental rate, are influenced by their environmental temperature. So you can see here that an insect reared in a cooler environment takes a longer time to develop than an insect reared in a warmer environment that will develop more quickly. And so therefore, I predict that bees reared at warmer temperatures will develop faster than bees reared at cooler temperatures. So the first objective of my study was to determine the actual temperature that developing offspring experience. And to do this, I measured the external air temperatures and internal nest temperature in sunny and shady locations. So here a picture of my sunny location this summer in my garden, and shady location in a tree, under a tree in my backyard. And I was able to construct what's called a dummy nest, where I drilled a hole into a raspberry cane to create an artificial tunnel. And because Ceratina don't reuse tunnels, there was no risk that they would take over that tunnel. So I didn't worry about stabbing them with the probe when I went to measure the temperature. But in any case, it was just a wire that I inserted into it about 10 centimeters down to record the internal nest temperature. And I had one of these dummy nests at both sites, one in the sun and one in the shade. And so the figure on this slide shows the date that I recorded their temperatures. So from late May to late August, which is the active flight period of Ceratina calcarata. And on the y-axis, you can see the maximum daily nest temperature in degrees Celsius, as was taken at 2 p.m. And we can see that in all but a few cases, the internal nest temperature in the sun was higher than that of the internal nest temperature in the shade. And this difference is significant with a paired t-test. And what we found agrees with previous research that sunny nests experience higher temperatures than shady nests. But what we have now shows that what we have now is internal nest temperatures, whereas the other studies, we only had air temperature. So now we know what temperature a developing bee can experience in a given summer. It's time to try rearing them to see how temperature affects their development. Without access to a laboratory this summer or an insect incubator, Dr. Adoni Skandalis had the brilliant idea to use an old Eppendorf Mastercycler gradient PCR machine that we had in our lab that was no longer suitable for molecular work. Here's the machine here, and then with the lid open. And what's great about the gradient function is that you can set the machine to express a temperature gradient across the whole surface. So you can have one set up to be cooler and one side set up to be warmer. And when you develop bees inside the machine, you can directly compare the effective temperature on the development time of the offspring that you use. So the second objective of my study was to observe how different temperatures affect offspring development time. So first I wanted to know what were the hottest and coldest possible temperatures that a developing bee might experience in its lifetime. So sorry, not in its lifetime, in its developmental time, so in the summer. And to accomplish this, I gathered historical temperature data from Environment and Climate Change Canada and their weather station in St. Catharines. And I searched for the hottest and coolest maximum daily temperature at 2 p.m. Using data from June, July, and August, spanning 2012 to 2019. Next, I used what I knew about the relationship between air and nest temperature to simulate the hottest and coolest possible nest temperatures a developing bee might experience. And then I programmed the BCR to simulate those temperatures. And I put some juvenile bees in it. And for the purpose of the analysis that I'm going to show you soon, I narrowed that analysis to prepupate and white-eyed pupae only. Recall that I gathered historical weather data for June to August from 2012 to 2019. This figure on the slide just shows the hourly temperature across the course of the day. And on the y-axis, we have the temperature. The red line represents the day with the hottest daily temperature that I found, and that corresponded to a date in 2016. And the black line is the maximum, so the day that had basically the maximum minimum, so the coldest maximum daily temperature, which was a date corresponding, which was a date in 2014. And then the gray line is the midpoint between those two dates. And I used this midpoint, to create the program for the BCR machine. So this figure shows maximum daily air temperature along the x-axis and maximum daily nest temperature along the y-axis. The orange line shows the relationship between air and nest temperature at the sunny location, and the blue line shows the same relationship in shady nests. And with a regression analysis, I determined that the effective air temperature on nest temperature is different between sun and shade. And what I mean by that is for each degree increase in air temperature, the nest temperature in the sun rises by 1.12 degrees, whereas it proportionally rises in the shade. So the figure on the left of this slide shows the temperature gradient by location in the BCR machine. So again, on the left-hand side, or in the case of this figure on the bottom, was the coolest temperature. And on the top of this image, or on the right side of the machine, was the warmest column. The figure on the right-hand side of this slide shows the hours of the day along the bottom, and again, temperature in degrees Celsius on the y-axis. The colored lines correspond with different temperature setting in the BCR. And using the midpoint line from my previous figure and a gradient setting of 10 degrees, the BCR was able to replicate a 20 degrees Celsius difference from column 1 to column 11. And with each column in succession experiencing a slightly warmer profile. The 24-hour pattern for all of the columns was the same, but only the range of temperatures were different. So bee offspring were placed in their own 0.5 millimeter microcentrifuge tube with the lid pierced to create some airflow. And the juvenile bees from sunny nests were placed on the warm side of the machine, and bees from shady nests were placed on the cool side of the machine. And I was able to open it and look through the tubes to assess their developmental stage about every 2 or 3 days. And this figure shows the BCR column in which the bee was placed on the x-axis, and on the y-axis the time it took to complete development in days. I have 23 bees, results for 23 bees that completed their development. And for the analysis, the bees were grouped according to whether they were placed on the cool side in the blue group here, or on the warm side, these red ones here. We can see bees placed on the warm side of the BCR develop significantly faster than bees placed on the cool side of the machine. And this result matches my prediction, but it also shows that the BCR is a viable way to study the relationship between temperature and development in small insects. To conclude, bees can survive inside the BCR machine long enough to complete development, and we saw that it's a viable solution to studying the effects of temperature on small insect development time. The added benefit of the BCR is that it can simulate the natural daily temperature fluctuations that occur in a nest. The BCR can also be used to investigate other temperature or physiological relationships, such as longevity, acclimation, thermal tolerance, metabolism, and temperature effects on body size. So I hope you all enjoyed my talk today. I look forward to answering some questions. I just wanted to give a quick thank you to my supervisor and my virtual lab mates who I miss seeing in person very much, and Dr. Skandalis for the brilliant idea. Thank you, everybody.
Yeah, thank you. There's a couple questions that we can get through before we get to the break. Jess Vickrook asks, why did you use large larvae or white-eyed pupae? Do you think you would have seen an even stronger effect if you used small larvae?
So I actually used all stages at first. When I first started opening the nests, whatever I found went into the machine just to see what could survive. I managed to hatch a couple eggs, and I found that the smaller larvae were particularly susceptible to whatever was happening in the machine, that they didn't often survive. So I found the highest survival rates with those stages. The earliest I could probably use was a fully grown larva. But I knew, for me, identifying white-eyed pupae was really easy, and so was identifying pre-pupae. So it gave us like the longest window of development, with also like a recognizable stage.
Yeah. And Nicholas Dorian asks, do you think that BCR would also work for early instar bees feeding on pollen? You probably answered this a little bit.
Yeah, I think if your purpose was to just rear them, then you could adjust the temperatures. Like I had very high temperatures on the right-hand side. If you changed the gradient so that you wouldn't have such extreme temperatures, it would probably be a good way to study the pollen body size sort of relationship, and maybe like consumption rates and things like that even too.
Okay. And Bandel Morrison asks, how many larvae did you start with? You said you ended up with data for 23 larvae, which completed development.
Yeah, so I had 23 calcarata, pre-pupae, and white-eyed pupae finished development. I started with 77 of all different stages, 1/3 to 2/3, 2/3 to 7/8 pollen ball size, eggs, what have you. Some of them, when they completed development, turned out to be Dupla McMackey, so they were excluded from the analysis that I showed you today. And some of them died early on as larvae. Some of them died at the body pigmentation stage. So it's what I ended up with.